I'd like to say that I'm very pleased to welcome all of you to the discussion today on virtual radiobiology laboratory. We have been discussing this for a long time, and it's really wonderful to see that the registrants have really spread across the globe from many countries, practically from all continents. So I hope everyone will be able to join by the time the discussion and the talks start. So we'll be looking forward to a very fruitful and output-oriented discussion. So just to give you a background, we have been working on collaborative projects between India and the United States for some time now, for about 2 or 3 years. And this collaboration had been initiated by Dr. Rath of the National Cancer Institute, All India Institute of Medical Sciences in Delhi, Dr. Badwe from Bombay, Dr. Jeff Buchsbaum and Dr. Noam Coleman from NCI US. And it has been very ably chaired by Dr. Bhatkar, who is the was the Chancellor of the Nalanda University and is also known as the father of Indian supercomputing. So this virtual radiobiology laboratory really is an offshoot and has emerged from the ideas that have developed in our collaborations in the past. In OHSL, and particularly Anil Srivastava, the president of OHSL, has been coordinating the efforts of the collaboration. And we would it would be really nice to see this widening and a greater scientific participation from different universities with different expertise, leading to several different kinds of proposals that could come out and collaborations that come out that would take forward the science of the radiobiology. So just to give you a brief brief background of who OHSL really is or what OHSL really is. Sachin, if you could give the first and the second slide, please. The second slide, please. So Open Health Systems Laboratory really has been built around the idea of team science or collaborative science. It was founded about 12 years ago by Anil Srivastava and Martin Deutsch. And basically, as it is written here, I'm not going to read out, this is there in the background for anyone to read, is to bring together expertise from different fields, different institutions, different parts of the world to address key challenges of life sciences. And the principle of OHSL really is that we believe that knowledge has the potential to contribute to the growth of science and empower people, only if it is shared and not restricted to silos. And this we accomplish by engaging participants, bringing participants together and to help them shape their own agenda for them to take forward their own agenda. Next slide, please. Next slide, Sachin, please. So the goals really is to carry out cutting-edge research in biomedical sciences and supercomputing technologies. These are the two major areas of work, particularly in biomedical sciences, particularly in the area of cancer. And going towards translational research and looking forward to significant improvements in health outcomes. And this we hope to achieve by building international teams around scientists, and mainly one of the major goals is to provide young and mid-career scientists an environment of mentorship and infrastructure and collaboration. So next slide, please. So this is just to say that recently we've signed an MOU with the Indian Institute of Technology, Delhi, for an International Center of Excellence for Computational Biomedical Sciences. This was signed between OHSL and IIT Delhi. So to move on to today's program, next slide, please. it's on virtual radiobiology laboratory, the VRBL that we shall call it. So we have 3 eminent speakers and 3 speakers with a lot of experience in collaborative science and collaborative research. We have Dr. Sandhya Shenoy from the Department of Biotechnology, the Ministry of Science and Technology, Government of India. And we are very grateful to have Dr. Sandhya Shenoy with us today. She'll be speaking on the Virtual National Cancer Institute and several other models of collaborative virtual institutes, centers. So she has been deeply involved in the development of planning virtual network centers for many diseases through the Department of Biotechnology. As Director, Medical Biotechnology and Program Officer for Chronic Disease Biology, she works on issues related to policy, planning and implementation. She's particularly responsible for driving programs in the areas of cancer, diabetes, chronic kidney disorders, autoimmune diseases, and many others. Dr. Shenoy has been closely involved also in the implementation of the virtual network centers in breast and oral cancers, cohort studies in Indian chronic kidney diseases, and SLE. So she will be walking us through many of these today, and we are really grateful for her knowledge and her long experience in developing such virtual consortia, which we will look forward to drawing from. And we are extremely grateful for the encouragement that we have received from DBT to take forward this endeavor. So I'll just briefly introduce all 3 speakers now so that we can just move smoothly into the talks without any interruption. So the next talk after Dr. Shenoy will be by Dr. Pawan Sharma, also a close associate and advisor of OHSL. His area of research has been in medical microbiology, particularly TB and malaria, molecular immunology and cell biology. So while at the International Center of Genetic Engineering and Biotechnology, Dr. Sharma had established a tuberculosis aerosol challenging facility and a first of its kind BSL-3 containment facility for tuberculosis research in India. He has published more than 100 research papers and in internationally peer-reviewed journals such as Immunity, PNS USA, Journal of Immunology, and many others. And what is pertinent to today's discussion is that he has established one of the first virtual centers of excellence for coordinated research in tuberculosis, which will help in guiding us through what we are trying to develop now. And we look forward to his talk also. And we have, of course, with us Dr. Jeff Buchsbaum, Program Director, Radiation Research Program at the National Cancer Institute. Dr. Buchsbaum has been a close associate, as I had mentioned earlier, with advising and collaborating on research ideas in radiobiology. He joined the radiation research program, clinical radiation oncology branch in 2016 at the NCI, and his various degrees include the MD PhD from Johns Hopkins and subsequent advanced training and faculty positions in pediatric oncology. He joined RRP NCI from Indiana University, where he was leader at the proton facility and recently promoted to professor. And he has to his credit 3 patents that were submitted for work related to proton therapy. So Dr. Buchsbaum is a fellow of the American Society of Radiation Oncology. He is on the editorial board of several leading scientific journals and a reviewer in many. He is also, of course, the recipient of many prestigious awards, among them the NCI Directors Award of Merit 2019. He will be walking us through the virtual radiobiology laboratory, the main area of discussion today, and basically giving the background to how this idea emerged, at least the initial plan for it, and how we can go forward. And we look forward to a good, stimulating discussion after this, and looking forward to many collaborations and at least scientific interaction on this. So over to Dr. Shenoy.
I'm going to briefly talk about the virtual network programs commissioned by DBT with respect to 3 diseases: cancer, chronic kidney disease, and an autoimmune disorder, systemic lupus erythematosus. Next slide, please. Virtual network centers facilitate centralized platform for sharing concepts and resources. It brings in complementarity expertise of individual PIs drawn from different organizational settings, creates diversity in size and complexity, helps to develop and actualize research agenda, and capitalizes on institutional research capabilities. There is no construction of building, there is no physical structure. It is a virtual networking of organizations, institutions, with a team science approach, where the project coordinator assembles a team to address basic, applied, translational, and clinical questions. Next slide, please. One of the established virtual network centers, VNCIs, is on breast cancer, entitled Multi-omics Analysis to Decipher Mechanisms of Hormone Resistance in Breast Cancer, implemented by DBT in 2017. There are 5 participating institutions in this VNCI. Tata Medical Hospital, which is the coordinating center of this VNC with clinical expertise. PI, the project coordinator, is Dr. Sudeep Buchsbaum, who is a clinical oncologist with expertise in breast cancer. Second institution is ACTRAC, with expertise in whole exome and transcriptome analysis. Third institution is National Institution of Biomedical Genomics, with expertise in whole genome analysis. The fourth one is National Center for Cell Sciences, on board for establishment of in vivo and in vitro cell lines and patient-derived xenograft models for drug screening. The last institution is Institution of Bioinformatics, where phosphoproteomic analysis of samples is being carried out. Next slide, please. Next slide, please. Next slide. Am I audible? Yeah. The next, yeah. The study envisages to understand diverse biological pathways of therapy systems at multiomics level to derive holistic understanding of mechanisms. As mentioned earlier, whole genome exome sequencing pertaining to point mutations, structural alterations, promoter region mutations, and germline are carried out both at NIBMG and ACTRAC. Whole transcriptome sequencing encompassing gene expression, transcript fusions, expressed variations, and mutations are being carried out at ACTRAC. Epigenetic modification, promoter methylation, correlation to gene expression is carried out at Institute of Bioinformatics, and establishment of patient-derived xenograft mouse models, in vivo and in vitro resistance mechanisms, validation of targets identified by other assays is now currently done at KIIT Bhubaneswar, as the PI from NCCS is now on superannuation, has moved to KIIT Bhubaneswar. Next slide, please. This slide depicts the layout of distribution of samples from TMC, TMH, which are surgically resected biopsy samples employed to establish PDX models. Blood samples are also collected to carry out genomic analysis and cell-free DNA analysis. Next slide, please. This is the governance model. PIs drawn from all 5 participating institutions constitute the working group that meets on quarterly basis to share the progress and status of sample distribution. Project also gets evaluated through different committees and mechanisms of DBT, DBT Technical Expert Committee, DBT STAG Committee, and Joint DBT Apex Board. At times, site visits are also organized based on the recommendations given by TEC and STAC. Next slide, please. Progress of the study thus far. Study target 390 patients, 195 resistant and 195 sensitive. 167 of the 390 patients have been recruited, 97 sensitive patients, 70 resistant patients, 128 samples distributed to other collaborating centers. Ongoing clinical follow-up of patients and additional patient recruitment is underway. To summarize, TMH has recruited 167 of the proposed 390 patient samples. ACTRAC is assaying and analyzing whole exome sequences. NIBNG is carrying out whole genome assays, and NCCS is establishing PDX models. We now move to the next study. Next slide, please. Indian chronic kidney disease. This is a prospective observational cohort study commissioned by DBT, present Indian chronic kidney disease, where subjects, the patients, are in early stage of chronic kidney disease. Next slide, please. This is a multi-centric study at 11 different geographical locations in India, with PGI Chandigarh as the coordinating center. The rest 10 centers include Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow, Institute of Medical Sciences, BHU, Institute of Postgraduate Medical Education and Research, Kolkata, Samarkand Kidney Institute and Research Center, Bhopal, Ujjibai Patel Urological Hospital, Nadia, Osmania Medical College, Hyderabad, Christian Medical College, Vellore, JIPMA, Puducherry, MNC Chennai, and Indira Gandhi Medical College, Shivler. The aim of the study was to understand progression of CKD, development of progression of cardiovascular disease into chronic kidney disease, variation of chronic kidney disease based on gender, development of predictive models, that is relationship between risk factors, and identify targets for intervention for future clinical trials. Next slide, please. The Indian chronic kidney disease kicked off as a pilot phase from 2013 to 2015, and then a multi-centric phase with 11 centers was conceived. It is managed by a governance structure comprising a steering committee with internal and external experts having regular meetings. The steering committee plans, executes, and assesses tasks to achieve objectives. Every center is self-sufficient, has trained manpower with basic lab facilities, and has experience in electronic database management. There has been a serial annual biobanking of samples such as urine, blood, and DNA at each center, which is then shipped to the central biobank that is being created at PGI Chandigarh. Next slide, please. ICKD study has enrolled 2,000 subjects across 11 centers. Follow-ups are still continuing. It is a national resource created by DBT. DBT has completely funded all activities at all 11 centers. It's one of its kind in developing countries. It's a part of the international network on chronic kidney disease. The data generated has become a resource for deciding policy and intervention. Next slide, please. This is their biobanking structure. Each center has got a local freezer. Serum, plasma, urine, and DNA samples are being stored annually. Samples are barcoded and electronically managed by ICKD database, and finally shipped to Central Biobank facility at PGI Chandigarh. Next slide, please. This is the outcome of the study. Notably, two important publications, design and methodology, has been published in nephrology. Control of hypertension in CKD across various cohorts, including ICKD, has been published in Kidney International Journal. In fact, this is a completed study, and DPT has very recently approved for the second phase for follow-up of the recruited ICKD cohort patients to study other parameters such as end-stage kidney failures, cardiovascular events, predictive models to identify high-risk subgroups. Next slide, please. Coming to the third study, this is again a cohort study in an autoimmune disorder, systemic lupus erythematosus, commissioned by DBT in September 2018. This has got 9 participating institutions, with SGPGI-MS, Sanjay Gandhi Institution of Postgraduate Medical Education, Lucknow being the coordinating center. The other 8 centers include All India Institute of Medical Sciences, New Delhi, PGI, MER, Kolkata, CMC, Vellore, St. John's Medical College, Bangalore, JIPMER, Puducherry, Nizam Institutes of Medical Sciences, Hyderabad, PGI, Chandigarh, and SCB Medical College, Cuttack. Next slide, please. This program envisages to understand the diversity of SLE. This is essentially an inception cohort from different parts of India for the assessment of clinical features, socio-demographic features, autoantibodies, disease progression, outcome, and evaluation of regional differences. This is again a multi-centric, multi-ethnic cohort where the patients are both pediatric and adult. This study also intends to create a biorepository to store samples that can be used for genetic studies and for biomarker identification. This cohort study has got 2 sub-studies, one on vitamin D, the other one on fever. Just like corporate social responsibility, this cohort has created what is called as clinical social responsibility under the name INSPIRE. This group carries out weekly patient education class on SLE. This is a WhatsApp group. There is a phone-based SLE helpline for patients, and there is a patient support group via WhatsApp that has been created to address all questions pertaining to SLE. Next slide, please. My talk has covered 3 diseases: cancer, kidney, and SLE. All the 3 programs are virtual networking centers where DBT has funded each center of the VNC. Each VNC described here has a project coordinator who takes the onus and drives the program. They have periodic group meetings to discuss the progress achieved. This is followed by periodic review by DBT through 3-tier mechanisms: technical expert committee, second one, a scientific and technical appraisal group, and third one, the ethics board. Thus, DBT keeps a tab on all the VNCs through these committees and review mechanisms. Thanks for your attention.
Thank you very much, Dr. Shenoy. We will be taking questions at the end of all 3 talks, and thank you very much, because it does give an idea of the different layers, the kind of collaborations, the methods to go about it, and I'm sure it was useful for a lot of us who are listening to it and looking forward to more such collaborative ventures. I now request Dr. Sharma to present his talk, and he will be talking about his experience of the Center of Excellence for Tuberculosis.
Okay, good morning, good evening, namaste to everyone. Am I audible properly? Hello?
Yeah. Yes, Dr. Sharma, you're audible perfectly.
Okay. Thank you, Sandhya, for bringing up-to-date information from DBT. We have come a long way from the very first virtual center which DBT supported. And I think I must congratulate Sandhya and DBT in general for laying down a very efficient framework for implementing such ambitious projects, which gives rise to, which gives opportunities to almost everyone who wants to take part in such studies from the country. So, and she has already set the stage, and I am expected to talk about how we started. So I think this is maybe this will be of more interest to those who are yet to embark on starting such a program. So maybe my presentation will give some kind of encouragement, hope, and direction. Although I would say this is 15 years old exercise, many things have changed, but the basic lessons which we learned, the way we went about putting together a program and so on, that I think that is what I'm going to share. Today, Sandhya presented in the very first slide, so there is a particular kind of attributes which define a virtual research center. But when we started, there was no attributes like this. We were just having our own ideas and so on and so forth. And I'll even go back from 2005 when we started this center. I will start from the TV program when it was initiated in ICKD in 2000, year 2000. I put there as it initiated from ground zero. Zero expertise. I was not a TB person. I didn't know even the T or B of TB. And then there were no facilities at all. So when Institute was given responsibility to start a TB program, the director picked me up. Most probably I was the most pliable scientist in the institute. He could say things to me, and of course I will of course have to take it. So I took it with a lot of ignorance because I had no idea what TB is, and also a bit of arrogance that yes, why can't we do it? We can do it. So everything happened, in fact, by way of these two factors, arrogance and ignorance. We started with cloning a small protein called MTSA10, just with the hope that nobody has studied this, because the paper which talked about MTSA10 was talking about ORF. And from my own simple knowledge, I knew that when you talk of ORF, probably you do not still have a real protein in hand, you are still talking about ORF. So maybe this is a good protein to start with, and we started with that. It was much later on I realized that this is very well-known protein called CFP10, cultural filtrate protein 10 or 10 kilodalton of Mycobacterium tuberculosis. And if I had searched Google, or at that time probably there was no Google PubMed at that time. If I had searched PubMed at that time, maybe 2,000 papers will come out of if I search the CFP10. But MTSA10, just one paper. So just to give you an idea that where we started. So even with that kind of, so since I starting with zero, I kind of got in touch with the TB experts from across the world and asked them for ideas, and whatever ideas I had, I shared with them. And I can't see my slide. So
yeah, your screen is here, Pawan. We all can see it.
Okay, I can't see it. So to do. Anyway, so I can open my thing here. Yeah. Okay. So previous slide, Sachin. I just want to run through that and then previous slide, please. Previous. Yeah. Okay. So try to get funding from Wellcome Trust and thinking that well, Wellcome Trust also funded me my travel to go to St. George's Hospital in London to work together with Dr. Phil Butcher there for about 10 days to put together a project. So we wrote a collaborative international research grant at that time. It wasn't funded. Wellcome Trust rejected it. And with two very good comments. The comments were that you don't have the expertise, you have no background in tuberculosis. But the more serious objection is that you also don't have any facilities to carry out any work related to handling live MTB. So you can imagine this. So this was a block there. So naturally, I had to tweak my project a little bit here and there, and we, within our limits, within my strengths and limits, I rewrote the project and submitted it to DBT, and there we were successful. In fact, the project was actually very much appreciated. Most critical person like Dr. Samir Brahmachari, he actually made the comment in the meeting itself in front of me that this is the most thought through project I have come across. So I knew that I'm going to get it. But it was not so easy. This is for information of Sandhya also. Sandhya, back then in 2001, ICGB will not get any funding from any national agency because we were considered international and we were multilateral ties and so on. So no funding agency will give. Mine was the first project which actually brought about a policy change in DBT. File went up to the ministry to allow funding of grants from national agencies to ICGAP. So that first project came. We started our work, and within a year or so, in 2002, first publications from my project, starting from zero, we came out with two publications: Infection and Immunity, Journal of Immunology. Meanwhile, we realized that Wellcome Trust had already said it very eloquently, and we also realized that how long, because we were only working with recombinant proteins and cell lines, that's it. No real MTB in hand. So there was need to put together a BSL-3 facility, so I started creating noise about it, and I was shocked to know that India actually didn't have any BSL-3 facility in any public institution in India. The only BSL-3 was in AstraZeneca facility at Bangalore at that time. I wrote to WHO. WHO was very gracious. They sent their expert to ICGAB, and they also had discussions with us. They gave me some money also. And with that money, we started training and all that. And I trained my people, trained myself in working in TSEF. Allowed me to travel to the US to 2-3 labs and to South Africa to 1 or 2 labs. So finally, the funding from DBT came for BSL-3, and that project was from 2005 to 2010. In between, at 2004, I had also organized one international TB symposium where a lot of people from all over the world, I mean, from NIH, from South Africa, Valerie Mizrahi, from NIH, Cliff Berry, and from Cornell University, and from, many people, so many people from all over the place. They came just like a keystone of India. So it was a mini keystone in India. So therefore, we had the right people, we had the facilities to come up, and now only thing was to think of big programs. So a lot of discussion started happening, and that's how the concept of coming together came up. And I would also say a catalytic factor was also at that time the secretary of DBT at that time. I want to pay tribute to the memory of Dr. Han at this moment. He also took a keen interest and brought many people together. so that we can put together a program. And he and the finance people at that time, you need two people. The top leadership sometimes gets handicapped because of the breaks put by the finance people in the departments. Anyway, so but at that time, two people, both the top leadership and the finance person, Mr. Pandian, together with this team was great team, and they started many initiatives which still are running, BIRAC, CDSA, and so on and so forth. So many initiatives they started. And they also came up with this idea, why not have a huge program? Not huge in the sense that many people should work together. TB is such a huge problem. It's not that one or two labs will work and find some solution. Why not bring together people so that you can work together and then attack the problem from different angles? And that's how the idea of virtual center for excellence came up. And we came together and we started making, writing up this project. And in the meantime, the facility was set up and it became functional in 2008. Supplementary funds also came for the facility in 2007. Next slide, please, Sachin. Next slide. As I said that we had, even with the simply recombinant proteins and the cell lines, we were able to show something, that these proteins, which people were actually at that time talking about them as a potential vaccine candidates, but we found that no, they can also work in a very different way. They can actually downregulate the inflammatory responses in macrophages and dendritic cells. So I think this is where we got our foot into the gate, so that we could get into the TB fraternity, although we had no background, nothing at that time. But we were talking about different language. We were not talking about vaccine at that time. We wanted, of course, we started with vaccine, but we found that these molecules are doing something different. So because we were talking different, I think we got the opportunity to get published in Infection Immunity. And then, of course, there was the Journal of Immunology, TC paper. And some papers came out just with the recombinant proteins and the cell lines. That's it. And so therefore, you again appreciate that there was the need for going beyond this model where all information we are generating is very artificial system, just the two recombinant protein and the cell lines. We need to go into the real model system. So that's why their facility was important. And that's why I think people from various disciplines were required to come together. Next slide, please. Next, please. So this is how conceptually, this is how we brought together people. So there was, at that time, we conceived that there will be a central MTB and tissue culture facility at ICGEB, which will receive very well characterized clinical samples, clinical isolates from Central Jalma Institute of Leprosy and other mycobacterial diseases, Agra. This is famous for Jalma, famous as Jalma Institute. We brought together people from University of Delhi South Campus to look at the genomics part. And ICGV will take up the proteomics part. My first grant was, we had done some proteomics work there. I haven't talked about that. And then, interestingly, this is where we went beyond our usual thing. We tried to involve colleges and universities who were not directly so much active in this thing, but they were good, say, for example, they were very good at chromatography, for example, fractionation and so on. So we will bring them on board. And similarly, Bioinformatics Center from JNU was also brought on board. So all these things, all these activities were being put together as Sandhya said, that a centralized platform where people will share their resources and share their expertise and ideas and responses and so on. And all of them complementary expertise, they came together. And so at ICGB, we were already working with macrophage cell lines. We were already trying to set up something on intracellular MTB growth patterns after we established the facility. And that's where we will also, we thought that we will study the drug sensitivity profiles and so on and so forth. And from the infected macrophages, the idea was to do the kinetic profiling of proteins which are being presented in context of MHC molecules very early in the infection. I think this is very critical point. Maybe I want to say a few things about it. I'll come back to it. And but more important thing here was to study the systems biology of MTB macrophage interactome from the host cell viewpoint. We were not doing too much of emphasis on MTB itself, but how the MTB modulates the macrophage. What are the systems of macrophage which are getting modulated so that MTB can survive there? So that part was, so it's very, very well defined, very separate thing. While University of Delhi South Campus, UDSC, they focused on generating auxotroph mutants, which they hope to use them as vaccines. Similarly, essential gene mutants, so that the genes which are considered essential, if they are mutated, how that thing will behave. They will, all these things, all these mutants will come back to ICGB for their study in the intracellular growth pattern and so on and so forth. And then, of course, functional studies in MTB H37 are about how these genes, essential genes, how do they really functionally, how important they are, and so on. So you can see that there is a emphasis on MTB here, there is emphasis on macrophage here, and both of these studies, they sort of complement each other. And the background information being generated on in silico analysis of essential genes. There was a list of essential genes which was created by Eric Rubin and Sassetti, Harvard, from Harvard, and that. So what are those genes doing? I mean, bioinformatically. I mean, just in silico analysis of those genes. What are the predicted structures, functions, and so on and so forth? Where are they located, and things like that. So, and then if we can clone and sort of validate those predictions experimentally, then of course we will go forward from there. There were also a component from the marine organisms. There has been at that time discussion about possible therapeutic molecules coming from the marine organisms. So National Institute of Oceanography, Goa, was of course put on board, and they did supply. I'll talk to you when I'll talk about it. Whatever worked, whatever didn't work. That's also important. So that of course was on board, but not very active. But but they did go through some fractionation. Some people got trained, and something happened there also, although not too much. And I'm not able to see here, so I see what is there in this panel here, animal studies. So whatever information we get from here to design a vaccine, or from here to design a vaccine, to come to the animals testing systems for final evaluation of those whatever molecules we think are going to become vaccines or maybe targets and so on. So this is the kind of virtual network kind of we created, and I must say that both these group here you can see JALMA contributed nicely by bringing together field isolates to the study. South Campus and ICGB did their job very well. This part remained little weak. We couldn't go too far into it. In this case also, we couldn't go too far into it. We did make some kind of progress. But I'm not too worried about that because a lot of progress was made in this case in the systems biology studies, which were kind of very novel at that time, and people were not even looking at MTB macrophage interaction at that level at that time. So these were probably the novel studies, and a lot of effort went into it. A lot of students were devoted to this, and so on and so forth. Similarly, these two components actually were very big components by themselves. And of course, the clinical interaction with the JALMA on one hand, and one part which is not written here, which is unwritten part, aims Dr. Sarman Singh's lab from All India Institute of Medical Sciences. They also provided us with some clinical samples and the clinical material, tissues and so on. So next slide, please. Next slide, please. So the overall approach was very simple. It was very sort of, the objectives were kind of, or the thrust of the program was really kept lean so that we can focus it. So a core program which was targeted toward discovery of drug targets and vaccine candidates, as I just explained, and a new model for high throughput research so that thinking that large data sets will come out from these studies. And so therefore, and which of course did come. So therefore that will help us to generate high throughput research. But some USP of this thing was strengthening national capabilities in TB research. The labs were accessible or kept open to anybody who wants to come and collaborate and work at ICGEB or at the University of Delhi South Campus, and so on. So, in fact, many colleges, they got benefit out of it. One of them is, of course, ANDC, where our Urmi Bajpai is there. Her students came to ICGEB for training for their small project work. So they were involved into cloning and expression of some of the genes and so on. They got trained in that. Similarly, there is a Shri Venkateshwara College, SV College in Delhi itself. So people came from there. Amity University's Department of Bacteria. So the idea is that we were not limited to our own institutes alone. The idea was to spread out so that we can recruit many more well-trained manpower so that this work can, or the TB expertise sort of, there should be a kind of what we call a critical mass of people who are able to make some difference. So this was the overall thrust of the. Next slide, please. Sachin, next slide, please. So core programs had only three. As I said, just three objectives: identification of MTB-derived antigens presented by infected macrophages in the early stages of infection, both MAC class 1 and MAC class 2. Develop a tuberculosis oxotroph with multiple gene disruption as a candidate vaccine. So this is the first objective is ICGB. Second objective is University of Delhi South Campus. And both objectives feed into each other. 1 and 2, the objectives, they feed into each other. And of course, a very ambitious program on systems biology approach towards understanding the molecular basis of pathogenesis. So, but from the viewpoint of macrophage rather than tuberculosis. So there is in the very first objective, there is a phrase on early stages of infection. I think there is a philosophically, we thought that, you see, if you look at the natural history of tuberculosis, almost everyone in India gets exposed, but not everyone comes down with disease. So which means although we get infected, but large number of people, very early in this stage of infection, they are able to mount a very strong immune response, which kind of controls the infection, but cannot eliminate it, because we know that later on, under any insult to immune system, these infections, they come up. So which means what is important is to identify those antigens which are being presented very early in the infection, to which the host is making a very strong response, but not strong enough to eliminate the infection. So if we know those identify those antigens and then study them in detail for their immunogenicity and so on, and protective efficacy, then we thought, I mean, this is all hypothesis arguments going on, that of course then there are strategies to make them strongly immunogenic, so that the immune responses which can be protective or which could eliminate MTB at that stage probably are possible. So I think this is so this was the idea behind looking at these presentation of antigens at early stages of infection, both MSC class 1 and class 2, in the context of both MSC class 1 and class 2. So this work was again done at ICGV. It has taken a long time. I think it is still going on. I mean, you may be surprised. I mean, I don't know if it's still going on, because those people have retired now. The point is, I'm not so sure, because I'm out of touch with literature for last few years. But I don't think there is any study on the kinetics of secretion of these antigens in MTB within macrophages. So we do not even know what is the kinetics, what kind of secretion levels are secreted, levels are there of these antigens at different stages of infection and so on. So I think there are still many unanswered questions in TB itself, I mean, despite all the work going on. And I believe that is important. Sometimes maybe just the level of a given protein may be important to skew a response one way or the other. So, and that probably needs to be investigated more thoroughly. I think I have given you a flavor of what. Next slide, please. What we were trying to put together. This project was, of course, funded for 5 years, I think. Yeah, from 2005 to 2010. It yielded about 22 publications. I can't see the figure here, maybe 22, 24.
Hello, Dr. Sharma.
Huh? Yes, I'm done. 15 PhDs and 3 patents. Some of the important publications are here: cross pathogens, cell, microinfection, nature, chemical biology, PLOS ONE, vaccine, and so on. Some of them are here. All this is published work. People can go through that. Thank you very much. And of course, this project was, the work was presented to DBT. DBT was gracious enough to grant another 5 years to this project. So, of course, I retired from that. I was not involved in the next phase. Thank you very much.
So you should be able to see me now. Sachin, I'm going to drag the ball. And then I'm going to share my screen. This is Jeff. Okay. Share screen.
Thank you, Dr. Sharma. This was very valuable, given the growth and evolution towards greater and greater complexity, which is only available through the collaboration and the capacity building also that happened along with it. Dr.
So
Buchsbaum.
you should see my screen now, and you should hear me. Please confirm.
Yeah.
So
Yes.
good morning, good afternoon, good evening, good today, good tomorrow, good yesterday to everyone on the call. My name is Jeff Buchsbaum, I was introduced, and I want to share with you thoughts. This is high level, and it reflects one of three public components of a project which I've been working on with leadership in India for a number of years, as Anil Srivastava outlined and Dr. Ray outlined. The first was the Ayurvedic Developmental Therapeutic Program, which has taken off quite well. The second is a Colossus project, which is looking to port GEONT4 to field programmable gate arrays and other supercomputing aspects, which is doing well. And then this is the third concept, and it's sort of being introduced to the public really today. Critical to these things is OHSL. They are partners in the overall process and have facilitated a tremendously efficient ability to communicate globally, and I'd like to thank them publicly. So the topic today is the Virtual Radiobiology Lab, and you just see pictures of different labs and mentors leading their troops, so to speak, their students. So background and purpose, radiation biology, why pick this? Well, it's a very focused area that is important for a lot of different fields right now. And it contacts what you see in the picture on the left, everything from radiation oncology to physics to immunology to cell biology. So essentially, radiation biology is a context of science that really transcends its own title. So the broad scope of radiobiology research that one envisions for the Virtual Radiobiology Lab requires and facilitates resource sharing and coordination across the collaborative team, across each country and across the globe. So it's a bridge. And it's rapid, it's cost effective, it's critical. So the idea of using the infrastructure can be done in a way that doesn't require huge amounts of resources. And primarily, collaboration and mentorship are the focuses of this idea. And the picture on the right sort of illustrates the concept in that you've got a bridge. The idea is to take everyone together across the concepts that are the science and the work. It's not about the individual, it's about the team. That has to be the focus of this work. And critical to that, frankly, is leadership, as we just heard from Dr. Sharma. He spent, and I'm giving him a compliment that was not at all planned. He spent at least a slide talking about his own personal limitations and such in a very humble fashion. And the humility he demonstrates is exactly the kind of the content leadership that you need to make this kind of project a success. So I really appreciate him being an exemplar in this context. So the next concept is membership, team building. Members should consist of individuals and teams with strong demonstrated interest in this field. So you don't want people that are just opportunists. You want people that have spent some time in the space, that have their battle scars and are in the position to contribute. The process and structure has to be developed with input from DBT and the Office of the Primary Scientific Advisor to India. And this is really, these ideas and thoughts have come about in that context, and that this VRBL will be something that is managed and led by the government of India. I speak privately in that context. I'm a US government employee. These thoughts have been shared with leadership in both countries. But at this point, the view of NCI is collaborative scientist, but knowing the leadership of this VRBL project will be headed and led by essentially India's government. And I took the pictures on the right to sort of demonstrate some of the things you have to think about. When you're leading, you got a lot of people depending on you, and that picture on the top right sort of demonstrates that. And the bottom right is how incredible collaboration can really achieve things. So what you see, I worked for a while around the Amish in the state of Pennsylvania, United States, and they had barn raisings. And I may not be actually perfectly accurate in my statement of dates and times, but historically they would put up a barn in a single day. And what's remarkable about that is the Amish don't use electricity in the process. So everything they do is powered by human muscle or sometimes animal muscle. And finally, the JNCI paper, you see, The Future of Radiobiology, which was senior authored by Eric Bernhardt, one of our former RRP colleagues, really outlines some of the needs and such of radiobiology at this point in time in the globe. So funding, the next concept to talk about. And just want to demonstrate an expensive gadget in the lab that it's always nice to have these columns and they always cost more than you want. So the early phase of a project like this, of a VRBL, would have to be able to function with little to no significant funding. So what governmental agencies want to do is we want to see established relationships and collaborations. You don't want the money to drive things. You want the science to drive the funding, not the funding to drive the science. So it's very important that people realize from the beginning the funding isn't necessarily going to be enormous. It's designed to initiate, but not to control and or establish. So it's a collaborative engine built on existing resources and investments. That's the key, is that people are using what they have more efficiently, not necessarily demanding new things, at least not in the beginning. Members are encouraged to seek grant funding from national and international sources. And as an example, we just had an RFA in the NCI for high LET radiobiology. It was open to the globe, and its 5-year total distribution of funds is $19 million. And so our goals inside the NCI are to facilitate these kinds of things through transparent, open opportunities to the global area of science. And I can't speak for the government of India, obviously, but the role of this VRBL would be to put people in positions and teams in positions to be, frankly, competitive in that context, in a global scientific grants funding context. Meetings and such, these are logistics. I don't think they're as important as the time would take. But essentially, the key is that people would meet, they would talk together, they would actually be mentally a team, not just figuratively a team, but people really would have to feel like they were true collaborators. There's a difference between writing a paper together after all the science is done and developing the hypotheses, the experiments, and working through the problems of the science together during the process. The latter is the approach that would be successful for an VRBL. Doing things together at the very last minute is not exactly something that would be constructive. And whoever is not muted, please mute. So later phases of funding, whoever's not muted, please mute. So members will continue to seek funds via domestic and international funding, and eventually there will be increasing amounts of support for logistical and communications glue and funding radiobiology research activities, training programs, collaboration, seed funding for grants and such. It's very important to understand that one of the major focuses of this is going to be mentorship and the next generation. I'll get to that a little bit later. So the science, and this is the bulk potentially of this conversation. I'm going to be very superficial, and I mean no disrespect to anyone in any particular institution or any focus of science. There's a lot in radiobiology. It touches almost everything I can think of. So this is just a demonstration of different planning approaches one can use in ion therapy, looking at Japanese versus European approaches versus physical dose versus differences. And we are at this point in time close, but we're not exactly predicting the exact same dose across the biology, even today in the most simple terms. So I'll get to that. So this slide is not meant to make anyone get a headache. There will not be a test or a quiz. You will not be required to know all the things on the right side of the screen. I'm saying that sarcastically, but it's meant to be a demonstration of the degree of science that's out there that can be looked at in the context of a VRBL. So the possible foci or areas of focus of the VRBL could include model systems. Now, what is a model system? It's cells or animal models that respect the geographic differences seen in cancer across the planet. So most of the models that exist, most of the PDXs that I'm aware of on the planet represent Western cancer patients. I don't think that there is large investment or capacity for these kinds of systems globally. And that's the kind of thing that has ample opportunity for expansion and scientific endeavor. Biomarkers. Absolutely critical that we understand as we deliver radiation what happens to our patients. There's a lot of opportunity to study that. The tumor microenvironment, again, a lot of opportunity. We're just barely beginning to understand how cells talk to each other, how the immune system may or may not respond based upon other conditions in the patient, et cetera. That leads to radioimmunology. Immunology has become a very large focus of science globally in radiation oncology and all forms of cancer therapy. The radiopharmaceutical therapy focus of the NCI is not to be understated. I'm of the opinion, and I share my opinion with many other people in NCI, that the delivery of radiopharmaceuticals to patients has enormous potential. They're targeted, they're radiation, they can be both beta and alpha in nature, and mixtures of these things and targeting of different sites in the body, and in particular dosimetry and biological dosimetry in this space, is an enormous frontier that is wide open for science, and I'm extremely excited about it overall. Combination therapy and the science of combination therapy is critical. We have 4 UO1 groups that have done some amazing work in the United States looking at CTEP agents. That's what they were funded to do. But there's enormous capacity to look at non-CTEP, CTEP being the NCI's division of early drug design and such, an early drug study. But we have the ability to look at different kinds of drugs in different contexts that may or may not have limitations based upon the domestic limitations in the United States within the drug and pharma world. In other words, you might be able to look at drugs that are off patent that are difficult to study in the same rigor as on-patent drugs because of issues that I'm not going to get into today. Sensitization and protectant science. We've recently published a number of papers in this space looking at the Small Business Initiative Research Grant Program in the NCI and different outputs there. It's a large area of opportunity. Other agents and technologies. This is just the tip of the iceberg. You've got data science opportunities. You've got supercomputing opportunities. You have digital twinning opportunities. We have interactions now with the Department of Energy in the United States looking at these kinds of things. There are supercomputing applications that can be taken to patients that makes the ability to treat a patient in a more individualized and accurate fashion, hopefully something that we see in the near term. So reproducibility and validation. This is a big deal, and it's not something that is typically discussed. And one of the primary, I believe, focuses of successful VRBL and all science going forward has to focus on this as something that is going to be, one, acknowledged as a problem, and two, mitigated as much as possible. There are a number of papers. Nature had a paper on this about scientific reproducibility in the recent era. This is a radiation-specific paper at the top left. And the cartoon on the bottom left, people have probably seen this or versions of this. I think you should be more explicit in step 2. There's a miracle. It's just been customized for cancer. This is a big deal. I remember as a graduate student, when I was told by my PhD advisor and lab chief, Jeremy Bird, go do this. And it's in a biochemistry journal somewhere from 1953, the method of purifying some protein. So I went to the library, I got the biochemistry journal, I read the method section, I did the process exactly as it was stated, and at the end of the process, I had the right thing, and it worked out. It was checked out by mass spec. It was the perfect product. If you go to a journal today and you try to do that today, the estimate is it's only likely to happen in maybe 20 to 30% of the time. And that's a real issue. Science is about sharing how you do things, not just about the result, but the process. So validation is crucial in all aspects of biomedical research. A VRBL would plan to leverage its unique resources to provide improved research data integrity broadly. Standard operating procedures and tools would be mandated to be shared globally so as to enhance all radiobiology research. Work in the VRBL would only be allowed using these kinds of standards, traceable radiation dosimetry, et cetera, et cetera, et cetera. Okay, I think this is important. I think I've said enough on it. Workforce, mentoring, critical, super critical. Whatever we do is irrelevant unless we provide the next generation the expertise and the people to do the next level of research. Otherwise, it doesn't matter how many Nature papers you have. It doesn't matter how many science papers you have if you have nobody to do the science after you're done. That's an opinion. I think it's important to say that. The NCI spends a huge amount of money on mentorship and training programs. And I know the government of India is very focused on this as well. I think this is a global issue, and radiation biology is under stress, to say the least. So the VRBL will be engaged in multiple programs, both internal and linked to other national and international programs, respective of each node, meaning the location of the collaboration, so as to promote the next generation of radiobiologists and radiation scientists. This is perhaps the most important mission of the VRBL, to nurture the world's radiobiologist network and grow the field, one that is most often seen as contracting in conversations and such. This expertise will be of value to those who study medical issues in multiple contexts, such as human space exploration. It's not just about cancer, it's about a lot of other things. I'll leave it there. Concurrent with the focus on radiobiology, the VRBL will support research programs in medical physics, radiation oncology, radiation therapy, those are the people that put the patient on the table and immobilize them, and radiation nursing. In other words, this is a component of a successful matrix of clinical delivery and clinical science. And that's it for my presentation. So I hope that was what you wanted. I'm going to stop sharing and give the ball back to Sachin once I figure out how to do it. Actually, I'm going to give it to Anil. Anil, you're in charge, or I'm trying to give you the ball. And I will stop there. Can you guys hear me?
Anil, you are on
Yes, yes.
So I hope I think that's all I have to say. Thank you very much for everyone's time.
Thank you very much. And I think all the three presentations actually focused on the theme, on the theme and experience, which can go into the design of VRBL. And thank you, Jeff, for a very good explanation of what VRBL should be. I think with that, we should open up the discussion because Koninika, I think, why don't you manage the question and answer if people have questions? Because
Yeah, I'll do. There is actually in the chat only one question from Dr. Rao. This is, I think, directed to Dr. Sharma. And this is about, are there studies in the tuberculosis relapse-related omics and in relationship to the reappearance? Dr. Sharma?
Yeah, what's the question? I didn't get it, please. Can you repeat the question?
Yeah. Are there studies in the TB relapse-related omics and in relationship to the reappearance?
What is that? Omics or COVID or what?
Omics, omics, O-M-I-C.
Okay. Yeah, so the relapse is another big problem in tuberculosis. How, this is, again, I would say that a very detailed study has to be done, not just on one time point or one focus and so on. Even within the same patient, the bacteria are at different levels of their growth in the different foci within the lung itself, in single lung itself. Cliff Barry has done very extensive work on this to say that there is no uniformity there. That's why I said that there is a need to do very systematic study on the kinetics of the, or you can say metabolomics these days, maybe. How the various pathways are actually coming in play at different stages of the infection. I think that is still to be done. I think we are still a long way from really finding out the real answers to these questions. That's what my feeling is.
So I got a question from someone I'm not going to identify because I think they don't want to be identified. But so the VRBL will be there to connect various groups. The answer to that, and to those on the phone call that are not from India proper, are yes. The leadership of India and the focus of this idea, and I'm not going to get into all the details, but there have been conversations at very high levels. was to set something up to sort of jumpstart the situation across multiple large clinical centers and more recently some areas of scientific expertise in the country of India. That being said, the context of this phone call demonstrates an enormous, to me, interest on the international community in radiation biology. And clearly, I'm at the NCI and I'm not sitting in India, I'm sitting in my basement due to COVID-19. But I think that what has to be stated to answer that question is that yes, collaboration and establishment of collaboration in the space of radiobiology would be something that would be a good thing for the world. And I think that, like a lot of other governments, at least my government, the US government, if we can say something, we make it public. And so I'm not going to answer questions that aren't public until it's able to be public. So I've said as much as I can today, but I would envision um, an opportunity set if the government of India wanted to move forward, where collaboration would be something encouraged on a global nature, and where the long-term goal of those collaborations would be to establish world-leading science to achieve funding from any number of opportunities globally. One of the strengths of that is that funding has a funny way of being up in some places and down in others in one country or across the globe. If you collaborate like this, your science program is more safe from ebbs and flows of funding opportunities as things happen, oftentimes out of your control in the government. So I'll leave it there, but I'm cautiously optimistic that this is a very I think this is a very important scientific endeavor. I think the government of India has demonstrated in the speakers today an enormous capacity to do virtual laboratories. And I think radiobiology is a very important topic that would be a very wonderful radiation virtual laboratory focus. I'll stop there.
There is a question from Dr. Chatterjee from TMC Kolkata. Really thrilled and looking forward to this long-awaited virtual institute. Are there plans for opening up possible collaboration funding through the virtual network within indigenous RCT?
I would defer to DBT on that one.
And I think I'll just add another question to this. This is from Arun Shankar saying, from Trivandrum, asking what is the next step and how do we take it forward?
Anil, I think it might be wise for you to give your perspective on that.
No, I was going to just say that. I think I'll let Sandhya join in and talk about it. But I think the next step, as Jeff's presentation quite easily put forward, is for all of us to identify our science and what we want to work together with whom, and develop a portfolio of projects and ideas, and collectively look at where the funding could be obtained. I mean, there are several sources of funding, and OHSL will definitely work with each group on that. And we are also at the same time trying to identify the funding. There might be funding coming for DBT has its own program that is there. Other foundations and other international agencies have their own program. European Union has a program. NCI, NIH, NCI have a regular grant program, which according to one of the presentations not long ago is almost $400 to $500 million a year that goes out to foreign collaborations. So basically, I think we have to start, as Jeff was saying, we have to start with the science. What is the science we are interested in, and how do we come together in a teams as such, and then sort of collectively help each other to find the funding. I mean, and I think I believe there is money around, and I think there is a lot of it. I can say, as OHSL, I can say that just as an observer, India's leadership, scientific leadership, has been very interested in promoting international collaboration with the participation of the Indian scientists as such. And DBT particularly has been a pioneer for quite some time in actually setting up such programs with different bilateral agencies. I'll let Sandhya speak to that because she can talk from internally. Sandhya, do you want to speak to that?
Yes. We had a proposal from TMH Kolkata on carbon ion radiation therapy. I think this was discussed in TED as well. The proposal has not gone further because we are waiting to extend the life of MOU between NCI, NIH, DBT, NCI, AIMS, Georgia campus. And B, I think, as I said a little while ago, that you're still looking for funding for TMH for the 200 crore differential. I think once we get this and it's all in place, DBT can go back and call back for a presentation from TMH and we are ready.
Perfect, madam. Thank you.
I think we all have to, my kind of urging to everybody is we all have to collectively work on science and work on finding funding for each other, not necessarily for our own projects. And we need to work with a cohesive, if there was a cohesive portfolio like VRBL, that means that we can collectively also engage the funding agencies. And I found the funding agencies very interested and willing to discuss in that kind of a setting. So find the funding. And OHSL is developing capabilities to help, especially the Indian scientists, who, with their help with developing proposals for submission to for international funding opportunity. And I think Dr. Pawan Sharma described something very interestingly, as he mentioned, that when he went to Wellcome Trust, they did not fund it eventually. They gave some money to develop it, but that also came, even their negative answers were very useful in further developing the project. So sometime it is not, I think we all have to make the effort on all of these things and to get support for it. And there is, so we'll have to work on it, all of us. It's part of the work that we have to do. It's not something given. So there's no funding agency who has come up and said, okay, we are going to fund VRBL. We are trying to create a community of interest, make it into a community of practice, and into several multiple proposals. For example, Bruce Fadigon is on the call, and I think Yogendra is on the call from CDAC. And both groups, the Topaz group and CDAC and others, and Sachin from OHSL, and all of us worked together to actually develop a proof of concept and demonstrate what could be done in acceleration of FPGA, using FPGA acceleration of Monte Carlo simulation, which has resulted in a paper. And now we are all working together to look at how do we find it, how do we find the funding, do the work further. So I think that's a part of the collaboration that has to go into. Any other questions?
There is one more from Lokesh Agarwal from NCI USA. This is a question for Dr. Shenoy. Regarding the TMC studies on studying drug-resistant studies on breast cancer, are there other tumor types that are also being investigated for drug-resistant studies? NCI USA has a DRSN, which is a drug resistance and sensitivity network looking at both heme malignancies and solid tumors, which is a cohort of 5 different institutions in USA. Maybe a good idea for collaboration.
So the program that we have funded at TMH Mumbai is only hormone-resistant breast cancer. I know the type.
Sandhya, your voice kind of got a little muffled, so could you just slowly repeat it again, your answer?
Am I audible now?
Yeah.
Yes.
Yeah, the question was, I think, on drug-resistant studies in breast cancer, are there any other tumor types? It's just hormone-resistant breast cancer with TMH Mumbai. The study is only dedicated for hormone-resistant breast cancer.
You faded again. The question is actually an extremely important question. NCI is a very broad place, and collaboration is a major focus of NCI science in general. Our role in NCI, frankly, is to make the scientists that we fund in the scientific field in general as successful as possible. So one of the roles with this radiobiology idea was to use this as a focus to establish opportunities and infrastructure and communication pathways, and then have that eventually grow out further. So this is exactly the kind of thing, just this WebEx, initiating possibly some interaction that we were hoping to do. So I agree with Anil that science should move forward. I think as things develop independently, having people develop relationships and seek opportunities for doing good science is always important. There are 1.3 billion people in India. They get a lot. They have cancer. We have cancer in the United States. The globe has a lot of cancer. There's a tremendous need for all of us to work together to achieve what we want for our patients and for the science as a whole. So I'm excited about this, and I've been the point person in NCI for this sort of process. But I certainly am not alone, and all of my NCI colleagues are of enormous value, and they know way more about most things than I do. So I'm humbled by my colleagues. So there's going to be a lot of opportunity for everyone. Over.
Thank you.
Actually, Koninika, if I can just interject,
Yeah.
I think Jeff used this very interesting metaphor of the barn raising, and I think that's something that is very appropriate to what we are trying to do. Is actually we are trying to put our collective effort to build that barn. I mean, that VRBL is that kind of thing. So we have to remember that it has to be our collective effort. That's what team science is about. It is not starting on the other end of it. I think Pushpa has Pushpa Tandon is on the call from NCI also. And Pushpa has some comment to make. Pushpa, do you want to?
Sure. Hi, this is Pushpa from NCI, the Cancer Imaging Program. And the Cancer Imaging Program has a network, the Quantitative Imaging Network, which is basically a network of institutions or investigators around the country. And it had investigators around the world that are not funded by NCI, but are interested in quantitative imaging, and they have their own funding, and that's more than 17 countries around the world. So that would be a good place to link in some ways, because that's what we are interested. The QIN is all about, to use the expertise of the different members to develop protocols and methods for quantitative imaging. So that's a main issue, is when you do imaging from different institutions, the protocols are not the same. And one image, when taken to another institution, they want to do imaging again, and that's such a waste of resources. So the whole idea is to develop methodology that could remove that bias and remove different, standardize the methods that are to be used and how you could do that. So I think in this regard, of course, we have a lot of radiation biology sort of work involved in it as well. So I think QIN would be a great place, or great network to be involved in this, to collaborate with, and I can talk to Jeff about it and take it from there. Yep.
Thank you, Pushpa. Thank you very much. And you actually did not mention that Tata Memorial and Harvard University worked on QIN together.
That's right. Yeah, we all
see that.
Right. We already have collaborations with Tata Memorial. They are associate members of the network, and with MIT and Harvard and MGH. We also have groups in London that are involved in it. So there, people from all over the world are collaborating and working on this.
Before I hand it back to Koninika, I just wanted to say two things. I believe, from what I know, I believe that the NCI, NIH has a tremendous amount of knowledge, and the willingness to share the knowledge is more important Yes, the money is always important, but the knowledge is very important for all of us who are working on that. So I'm hoping there's a knowledge partnership that will continue and thing. So that was one part of it. The second thing is, as Pushpa mentioned about the quantitative imaging network, actually the OHSL's role, and one of my colleagues is here on the things, Sachin Dev, who actually assisted all the people involved to be able to carry forward what they were wanting to do. So, I mean, so whether it was with the software, with it connecting to it. So that is the role that OHSL plays quite often, is facilitating the scientists coming together and collaborating. And that is the important part of it. And I hope that we will continue with that. Yeah, Koninika, back to you.
Just a moment, and I have to mention that Anil has been very, helpful in getting collaboration set up, especially with Tata, with the QIN. Hats off, because without his help, we would not have been able to do it.
Thank you, Dr. Tandon. Anil, would you like to say something?
I was handing it back to you. I think we should conclude. We have gone.
Yeah, I think we have completed this meeting within the time specified, which is very good. And I just wanted to mention that this is really the start of the discussions, and there will be a lot of opportunities to get together either through email or through more virtual platforms like this, or we will also have a site on the web with the required privacy settings, so that we can have a discussion to continue on what kind of collaborations that can come about, what kind of research questions need to be asked, and to advance the science of radiobiology. And that is what is really needed. The discussions really will lead to the collaborations. So with this, I'm going to, I just want to thank everyone who has joined in and who have participated in this meeting. It's very encouraging to see you all over here. And I would also like to thank of course, not also, and the very important part of it, to thank all the speakers and to show their experiences, for Dr. Buchsbaum to walk us through the requirements and what we need to have in our minds to go in for the collaboration, Dr. Shenoy to show what DBT has done in the past and their experiences and how it has, and of course, Dr. Sharma's initial evolution and growth of the virtual network. So all this has been very useful and informative. And I would like to, at the end, thank our backend team who have been tirelessly working and who have got this meeting going there. They've been sending out the questionnaires, they've been sending out the announcements, they've been arranging for all the connectivity requirements. So this is Ayan and Ayan and his team, and Pulkit and Sachin, of course, and Anil and Sachin organizing this meeting, coming together. So thank you, everyone. And I think we will close here.
I just want to add one word. I mean, in a very chaotic fashion, I brought these three speakers together. And just as a listener to this, I realized that there was such a coherence between the three experiences that it was quite, for me, it was very gratifying. I think what we learned from Pawan is the process and the difficulty and how it all came together. I mean, and how it worked further. I think the fact from DBT, the experience of funding virtual laboratory projects and work of them, and that's very, very inspiring. And of course, as usual, Jeff has brought 2 or 3 years of interaction with the scientists on both sides in a very succinct form, and actually he has provided us a kind of I would say survival guide, if I can say, for the VRBL, that okay, how we need to plan our thing further and how to survive a complex thing like collaboration across the oceans and across the world. So I'm hoping that this will result in several collaborative projects. And we will try to see how we can keep in touch with this community that we brought together and provide information and help and work and take it forward. Thank you very much for everybody joining the call.